This is the first part of a two-part video series on cloning and stem cells, which are two very closely interrelated topics. In this part, we will discuss how mammals are cloned and the difference between reproductive cloning and a procedure called therapeutic cloning. We all start out life as a single fertilized egg cell. All animals start out life that way. And then this single fertilized egg cell undergoes lots of mitotic divisions and then produces a complex multicellular organisms where the cells look and behave very differently from each other. One of the fundamental questions of developmental biology then is what happens in this process of generating a complex multicellular organism? Do the cells in our body that make different organs and different tissues, do they undergo changes in their DNA, or do they undergo other changes? And so one of the ways to address this question back in the early 20th century was to try to clone animals at different stages of development. And they worked out this procedure using frogs, where it's easy to get at their eggs and their eggs are easily manipulated. So you take frog eggs that have not been fertilized yet and you remove their pronucleus. So then you have this empty egg cell, and then you can take nuclei from cells from an early stage embryo, or from later stage embryos, or from a tadpole. So the embryos in an early, or the cells in an early stage embryo, it was known that these cells are still capable of producing an entire organism. These cells are undifferentiated. They have not yet become different from each other, and they haven't yet committed to any particular pathway of differentiation. So when nuclei are taken from early embryo cells, and then these nuclei are transplanted into this empty egg cell, and then the egg cell is activated and tricked into thinking it's been fertilized and developing. So then the egg cell divides, undergoes its mitotic divisions, and what we see is that you get, or you can get, a normal looking tadpole. So the conclusion then is that the nuclei in these early stage embryos have undergone no changes at this point from a fertilized egg cell and are perfectly capable of directing the development of an entire organism. However, if you do the same experiment and this time take the nucleus from an intestinal cell, so this is fully differentiated, it's an intestine, it's not any other part, so the nucleus from intestinal cell, and do the exact same experiment, transfer the nucleus into an empty egg cell, and do the same activation and tricking to divide and undergo embryonic development. At first, it almost looks like a normal embryo, undergoes divisions and mitotic divisions, but then what we get is an arrested development. It goes to, in this case, what's called the blastula stage embryo, and then stops. No further development occurs. So this says that something has changed between the early stage embryo and even a juvenile tadpole, that in the process of cellular differentiation, something has changed in the nucleus of these cells. Is it an irreversible permanent change with involving changes in the DNA sequence, or is it other changes that we don't fully understand? So later on, as scientists were able to actually study the DNA sequences of cells, then they discovered that there were actually no changes in the DNA sequences of differentiated cells. So the process of cell differentiation did not involve changes in DNA sequence, but instead it involved other changes, maybe in the way that DNA sequences are regulated. So it appeared to be more a question of regulation of gene expression and how the nucleus is able to direct the use of particular genes to achieve cell differentiation. And then the question became whether that could be somehow reprogrammed. It appeared to be impossible to reprogram the nuclei of differentiated cells, in vertebrates anyway, although it appeared to be completely possible because we could clone plants fairly easily. But with vertebrate animals, it appeared to be pretty impossible until we were surprised in the late 20th century by Wilmut, who presented us with a cloned sheep named Dolly. So here's a picture of Dolly and her surrogate mother, and you can see that her surrogate mother is a black-faced sheep and Dolly is not. And Dolly herself was fertile, and she gave birth to a lamb named Bonnie by natural means, meaning she mated with a male, and Bonnie is a naturally born lamb. So how was Wilmut able to do this? He essentially used a very similar procedure as had been used for frogs. He took eggs from sheep, so he took sheep eggs and again enucleated them, took out the nucleus from the sheep egg to produce an empty sheep egg. Now he took udder cells, mammary cells, from a sheep of a different genotype. Okay, this is a fin dorset, and he cultured these mammary cells. And what he found was that the trick that he used was to starve these cells growth factors. So normally to culture these cells, you have to put them in medium containing not only nutrients, but lots of growth factors, provided usually in the form of serum. And if you take away the serum, it takes away the growth factors, which provide growth signals required for the cells to divide. So in the absence of growth signals, these cells go into cell cycle arrest. And the cell cycle arrested cells somehow are able to achieve some degree of nuclear reprogramming. Because then he was able to take the nuclei from these cell cycle arrested cells and put them into the enucleated egg. He had to apply an electrical pulse to cause nuclear fusion, or fusion of this nucleus with the actual egg cell, and this actually gets the nucleus into the egg cell, and then the egg cell begins to divide as if it was a normal fertilized embryo. And so you get in vitro development to an early stage blastocyst embryo, and then this blastocyst stage embryo is implanted into the uterus of a surrogate ewe, okay, a surrogate female sheep. And if that successfully implants into the uterus, you get a cloned sheep born. Okay. This is a little picture of the somatic nuclear transfer This is the egg cell here with a holding pipette to hold it in place, and this is the microinjection pipette, which is actually injecting the nucleus from a somatic cell. I want to stress that cloning of mammals in particular became possible because of the developments in human reproductive technology. This in vitro fertilization clinics really, or the in vitro fertilization technology began in the 1970s. Louise Brown was the first test tube baby, and when she was born, there was actually a great deal of controversy about in vitro fertilization and whether this violated religious and ethical principles. Well, nowadays, in vitro fertilization clinics are fairly widespread throughout the country and around the world, and lots of people take advantage of in vitro fertilization technology to have children. In vitro fertilization technology was also responsible, or human reproductive medicine was also responsible for developing the somatic nuclear transfer technology. So men with defective sperm could not produce sperm that was capable of in vitro fertilization, and to get around that, they developed the somatic cell nuclear transfer technology, taking nuclei from sperm and directly transferring it into human eggs. And then there were other technologies. So the ability to transfer embryos into the uterus has been used not only for human in vitro fertilization, but for reproduction of livestock, especially cattle. So all of these technologies were really made mammalian cloning possible. Now, the downside of animal cloning, or mammalian cloning in particular, is that it is still an inefficient procedure. To produce Dolly, Ian Wilmut tried 277 times before he was successful with Dolly. So he took 277 eggs and did 277 somatic cell nuclear transfer experiments. Only a relatively small percentage actually started dividing after the somatic cell nuclear transfer. Only a few embryos appeared healthy enough to transplant into the uterus, and then even among those uterus transplants, only one was successful to produce Dolly. So 1 out of 277. Nevertheless, many animal species have been cloned, including primates, cats and dogs, and of particular note is cloning of a mule. So mules are sterile because they're hybrids between donkeys and horses, and the only way really then for mules to reproduce would be by cloning. One question was whether cloned animals would actually be healthy. So Dolly was healthy for the first few years of her life, and she was a fully functional female because she was able to give birth naturally to a healthy lamb. But Dolly herself suffered from premature aging symptoms. She got arthritis at a much earlier age than would be expected for sheep of her genotype, of her breed, and as well as other ailments of old age, and eventually had to be put down. And a cytological examination and genetic examination of Dolly's chromosomes discovered that the ends of her chromosomes were much shorter than usual for sheep of her breed and comparable age. So telomeres are the ends of chromosomes, and there are special sequences at the ends of chromosomes. And these ends of chromosomes cannot be replicated, the telomeres cannot be replicated through cell cycles. There's a special enzyme called telomerase that in the germline simply adds telomeric DNA sequences to the ends of chromosomes to maintain the chromosomes. And stem cells and cancer cells express the enzyme telomerase, again, to maintain chromosomes at full length. But in normal somatic cells, as they divide, they do not express telomeres, and the chromosomes shrink with each round of cell division. Since Dolly's donor nucleus was derived from an adult somatic cell, we would surmise then that her telomeres to begin with were already shorter, and when they were introduced into the recipient egg cell. And then as Dolly's developed, her telomeres continued to shrink, and so that may be at least in part responsible for her symptoms of premature aging. Experience with reproductive cloning applied to lots of other model organisms and especially to livestock shows that there are some general and very common problems with reproductively cloned animals. And this has been given the name cloned offspring syndrome. Reproductive cloning leads to placental abnormalities in the surrogate mothers. Remember, cloning always requires a mother. You have to have a uterus to implant these cloned embryos. The fetuses have a tendency to grow too large, again causing problems for the surrogate mothers. There's a much higher rate of spontaneous abortions for cloned fetuses and early death. Okay, so premature aging like Dolly, or even earlier death, okay? Perinatal, shortly after birth. Okay? So shorter telomeres appears to be a general problem, but the problem that's causing all of these placental and fetal growth abnormalities appears to be based on a phenomenon called genomic imprinting. So mammals have genes that control placental growth and fetal growth, and these genes are marked in a special way so that in sperm the genes to drive fetal growth are on, whereas in eggs the genes that drive fetal growth are turned off. And this balance and this achieves the correct balance to produce an optimal rate of fetal growth, not too big, not too small. And in these cloned embryos, that imprinting doesn't work, or doesn't have the right balance. What we end up is an imprinting pattern that looks like two copies of sperm driving too high a rate of fetal growth and too high a rate of placental growth. So what we talked about is a procedure called reproductive cloning. So this is a figure, again, reproduced from earlier on in this video, where you go through the procedure, form these early stage embryos, and for reproductive cloning you would put them into a surrogate mother. Therapeutic cloning does not put them into a surrogate mother. Instead, therapeutic cloning takes these early stage embryos and destroys them, disrupts them, and collects a group of cells called cells from the inner cell mass, because these inner cell mass cells are pluripotent, and we will talk about that in the next video. And these inner cell mass cells are cultured in cells to derive embryonic stem cells. So therapeutic cloning means deriving embryonic stem cell cultures from cloned embryos. And that is the topic of our next video.